we are going to perform the estimation of blood urea levels. For that, we will be using a urease enzyme method. It contains enzyme, chromogen, and standard concentration of urea. And here are my patient samples. So let's start the test. We will be taking 3 test tubes and we will be marking first one as the reagent blank. I am marking it as RB. Second test tube for the standard, I am marking it as S. And the third test tube for the patient sample for the test we are going to see, I am marking it as T. Now we will be adding 1000 microliter of enzyme in all the 3 test tubes. We will be adding standard, which is of 40 milligram per cent in the standard test tube. We will be taking 10 microliter of the standard and we are adding in this standard test tube. And for the test sample, we are adding the patient's serum. Again, we are adding the same 10 microliter, third test tube, and mix it well. So after mixing, we have to keep this 3 test tubes for incubation period of 3 minutes. So now we'll be incubating this at 37 degrees Celsius for 3 minutes. Now 3 minutes got over, so we'll take the sample. After 3 minutes incubation, now we will be adding chromogen, 1000 microliter in all the 3 test tubes. After adding chromogen, we have to incubate it at 37 degrees Celsius for 5 minutes. After adding chromogen, we have to incubate it at 37 degrees Celsius for 5 minutes. Now the 5 minutes got over. Now we will be taking out the samples. Now we have taken the test tubes out from the incubator. We can see the color change. Now we can start taking the readings from the calorimeter. I am switching on it. setting the wavelength to 580 nanometers. So once we set the 580 nanometers, we will keep the distilled water blank to set it to auto zero. I am wiping it because of the excess fingerprints should not be there while taking OD. Now it has come to 0. Now we can start taking the readings. First I am taking the reading of our reagent blank. It's coming as 0.09. Then we will take the reading of the standard. It's coming as 0.27. At last we will take the reading of the test. It's coming as 0.02. Now we can see how to calculate the concentration. Now we have taken out the test tubes from the incubator and we can see the reaction color change. This thing will be measuring the OD in the calorimetry. I am switching on the calorimeter and I have set the wavelength to 580. So after setting the wavelength, we have to set the OD as 0 using distilled water blank. And before keeping the cuvette, I will wipe the fingerprints and other dirt particles. And this is automatic machine, so I have pressed the auto zero. Now the 0 has been set. Next I am taking the OD of the reagent blank. It is coming as 0.10. Next I am going to take the OD of the standard. It is coming as 0.27. At last I will be taking the OD of the test. It is coming as 0.22. So we have written all the OD values, and we also know the concentration of the standard is 40 mg/%. Now the formula for calculating the concentration, unknown concentration, is OD of the test minus OD of the reagent blank upon OD of the standard minus OD of the reagent blank into concentration of the standard. Now we have to just put the values and calculate. OD of the test is 0.22 minus OD of blank was 0.10 upon OD of standard is 0.27 minus OD of again the reagent blank is the same 0.10 into the concentration of standard is 40. On calculating this, we found out it's 0.12 upon 0.17 into 40. On further calculating, we get the final concentration as 28.2 mg/dL. This is the concentration of the test sample.